Group 10, and it's me, Pooja Casey, and our biosynthetic project is on converting pineapple waste into protein rich animal feed by engineering Saccharomyces cerevisiae. And thank you for joining us. As we all know, Australia is a significant pineapple producer, growing over 1 lakh tons annually. But with this comes a major challenge: pineapple waste. Large volume of peels, cores, and crowns are discarded, as seen in Queensland, where a single 100-ton loss cost nearly $3 lakh. Globally, the issue is even larger. About 15 million tons of pineapple waste are generated each year from 30 million tons of fruit. This is part of a broader agricultural waste problem in Australia, costing up to 36 billion annually. Pineapple waste is rich in sugar but low in protein, and it spoils within 2 days, making immediate reuse difficult. Meanwhile, the world needs over 300 million tons of plant protein for animal feed each year. Addressing this dual challenge aligns with the UN Sustainable Development Goals, especially SDG 12, reducing food waste, and SDG 2, improving sustainable food supply. Next, please.
Hi, my name is Tahsin, and I'm gonna talk about the methodology, where here we constructed the genetic design, and then we used promoter and terminator for engineering, and also later on we did the assembly strategy and verification, as well as documentation and version control and biosafety and regulatory control. I'm going to talk about the advantages that you can see in the figure. This system offers several advantages over traditional waste management and protein production methods. If I talk about the environmental advantages, it reduces landfill use and greenhouse gas emissions. And yeast grows, as everyone knows, yeast grows very rapidly, doubling in just 2 to 4 hours, and its yields are very high. And in terms of economic, we can convert low value waste, less than $10, 10 Australian dollar per ton, into a product potentially worth over $500 Australian dollar per ton, revealing imported soy meal. Now, if we talk about the nutritional factor, yeast protein is rich in lysine and vitamins and is highly desirable. Compared to black's soldier fly larva, yeast grows faster and does not require post-processing. Unlike composting, this method produces a unsuitable protein product. However, there are challenges like GMO regulations, contamination risk in sugary waste, energy cost for drying and aeration, and the need for fermenters and sterilization equipment. Now, the next will be presented by my friend Marcia.
Thank you. This is Marzia. Our genetic construct is robust, built on a high copy plasmid to ensure strong enzyme expression. We have incorporated specific genes like CBHA, PELA, and PEP4 under strong promoters to efficiently break down cellulose, pectin, and proteins from the pineapple waste. In essence, we are turning a significant agricultural waste problem into a sustainable solution for protein production, fostering a circular bioeconomy and contributing to a more sustainable future. Our engineered Saccharomyces cerevisiae uses a modular design where each gene, CHA, PLA, and PEP4, is under control of strong yeast promoter ADH1 and directed for secretion using an alpha mating factor signal. Each of these enzymes plays a specific role. Cellulose breaks down fibrous cellulose, pectinase targets pectin from fruit's wall, and protease degrades leftover protein. Together, this trifunctional system transforms complex pineapple waste into digestible nutrients then that yeast can then convert into microbial protein. These designs mimics consolidate bioprocessing, reducing external enzyme requirements and making it more cost efficient for animal feed production. We visualize these both a circular plasmid map and a linear cassette to clearly show how each module is assembled and functions. This clarity is essence for troubleshooting and future scale up. So here we can look that the three cassettes that are performing TEF1, which is a promoter, alpha factor secretion signal, TEF1 terminator, drives secretion of downstream proteins. Cassette 2, TEF1 promoter, PLA gene, which is pectinase, and PEP4 gene, protease. Here the terminator is TEF1 and enables to breakdown of pectin and proteins. The cassette 3 performs TEF1 promoter CBHA gene, which is cellobiohydrolase. TEF1 is same as terminator, breaks down cellulose into fermentable sugars. All coding sequences are codon optimized for Saccharomyces cerevisiae, and the alpha factor signal ensure enzymes are secreted outside the cell of efficient waste degradation. Thank you. Next speaker, please.
Engineering Saccharomyces cerevisiae to Thank you, Margia. This is Susmita here. I'm talking about the assembly and verification strategy for engineering Saccharomyces cerevisiae to convert the pineapple paste into the microbial protein. For the initial lab test, we used plasmid-based expression with PRS-426 for high gene doses. For the industrial use, we can integrate these cassettes into the yeast genome using CRISPR-Cas9 to avoid plasmid maintenance. We assemble the construct using Golden Gate assembly, a modular method with unique BSAI sites and custom overhangs. For scaleless and directional cloning, the verification includes the colony PCR, restriction digestion, and Sanger sequencing. The plasmid named PRS-426 PEL-A PEP4 CBHA is the first transformed into E. coli for the propagation, then into a URA3 delta S cerevisiae strain for the expression. Biosafety is the crucial, so we can use an auxotropic marker named as URA3 from which the yeast cannot survive outside the lab condition. All of the work compiles with Australian GMO regulation with low environmental risk due to minimal gene transfer and none of the pathogenic traits into the introduced genes. Next, please.
Samin.
So hello everyone, my name is Samin and I'll talk about the testing and the scale-up strategy of our experiment. So as you can see, our goal is to validate the Saccharomyces cerevisiae engineered using synthetic biology, if it can convert the protein, the waste into the protein. And the method we're going to use is we're going to conduct small-scale flask experiment using our engineered yeast and convert it into the pineapple waste with the pineapple hydroxylate, which is sugar and nitrogen rich. So meanwhile, we'll do our experiment. We'll monitor the cell growth and the optical density, protein output using the Bradford or BCA assays, and the protein quality by using the SDS page analysis. The laboratory outcomes that we're expecting are the high protein concentration in the biomass, strong growth patterns, and efficient conversion of pineapple sugars into cellular proteins. Next slide, please. So for our experiment, we'll start with the 1 to 10 liter the 1 to 10 liter bioreactor, which is the small one, to stimulate the scale production with continuous input of the pineapple waste slurry, and then we'll gradually increase our quantity to up to 1,000-liter tank when we move into the field. And while we're doing all this, we have to make sure we are following the optimized conditions like fermentation parameters, pH, oxygenation, stirring speed, and the temperature. For the livestock feed trials, we'll incorporate the protein-rich biomass into animal feed. And next, we can collaborate with the Agricultural Research Center to measure all the outcomes. And in the real path, in the real world, we're going to partner with some, big industries like in Queensland and animal feed manufacturers, evaluate practically the modular bioreactors deployed directly at the farms or processing sites, and conduct the life cycle analysis. So in summary of our project, we offer a sustainable, scalable way to transform pineapple waste into valuable protein, supporting food security and circular bioeconomy. So that was all from our side. I hope you enjoy and like our presentation.